Hello, and welcome to this edition of How to Study for Dr. Lawson's lectures. This lecture is on mutation and repair. It's not actually a lecture, as you can see from the front slide. It's actually something you have to study on your own, and then we work on the skill set required for this material in an interactive session. Each of these sections, of which there are 4, there are some basic questions that are being asked in that section. They are not necessarily the only questions you need to know, but they do give you a guide of what you need to be aware of. For example, in section 1, we need to know what are the causes of mutations. Do not memorize the cause of mutations. Instead, know the general gist of how you get mutations. This is probably the major part of section 1 that is important. What happens to the DNA if it's not repaired, and additionally, what kind of repair system does repair it. All DNA damage does cause mutations. Many of the mutations are single base pair mutations. We will see a lot of ways of making single base pair mutations as well as multiple base pair mutations. This is just simply a reminder of the nomenclature. We call a single base change a single nucleotide polymorphism, or an SNP. These are just single base pair changes. These alterations can be the kind that we talked about in the translation lecture. From Dr. Larson's lecture on chromosomal abnormalities, you will have been made familiar with a number of these types of chromosomal rearrangement. Now we're actually getting into some of the causes of mutations. The important thing to know here that if we make a covalent change to the base, each of these has a specific way of being corrected. Single base pair changes are called mismatches. Although I tell you in each of these closer look slides how the mutation occurs, that is less important to you. What is more important is how is it remedied and what happens if it is not repaired. In section 2, we talk about what mutations are, how we classify mutations. Why do they rarely cause disease? We get a lot of mutation that has no effect at all on our disease state. In fact, most mutations are completely silent because they do not happen in a protein coding sequence or damage the regulatory region around a gene. Lastly, we'll talk about which places in the genome we have the most mutations that cause disease. And of course, that's going to be in the transcriptional region for messenger RNAs, particularly in the exons of messenger RNAs. Most mutations in this spliced out introns has little effect. Splicing, we've already talked a little bit about. You know that there is a canonical splicing sequence. This slide goes into pretty great detail on what happens when you add splice sites or remove splice sites. You don't really need to memorize this if you just use the logic. If I know where that splice site is supposed to be, right here at the end of the exon, and I put a new one either inside the exon or outside the exon, you can use basic logic to figure out what that's going to do to the protein that is made from this gene product. That's really all this slide is telling you. So use your logic. Do not memorize this slide. I need you to very clearly understand this slide. The key point of this slide is this bullet right here and this bullet right here. In older parents, as parents age, they accumulate more and more point mutations with age. In men, it is point mutations that they accumulate because their sperm are going through many mitotic divisions. The sperm of a 50-year-old man has gone through nearly 1,000 mitotic events. For older women, the problem is not in point mutations because eggs do not divide multiple times after birth. In fact, they don't divide again until they are fertilized. So the problem with women is a recombination effect. So if the egg is fertilized and there's been a recombination event going on and it gets abruptly stopped or the DNA gets broken because of the recombination event, then in fact you will get large effects on the DNA like chromosomal rearrangements, large deletions, large insertions, translocations. This is a slide to remind you of the different types of point mutations. You can have silent, missense, nonsense, as well as you can have insertions and deletions of either a single base, or you can have multiple bases if the insertions and deletions are more than a multiple of 3 or less than a multiple of 3, then the mutation will cause a frame shift. Starting at the frame shift, the amino acid sequence will be changed, and typically this results in truncated proteins because there are typically many stop codons in each new frame. This is the most important part of the whole lecture. This is section 3. It is what are the types of repair systems and when are they used. I strongly recommend that you make yourself a little table of the various repair systems and you figure out which kind of lesions they fix and which kind of lesions they cannot fix and what kinds of diseases are caused by the repair system not working. So what are the repair systems? There is the one that happens during DNA polymerization, which is DNA polymerase proofreading. Remember, only DNA polymerase delta and epsilon can do DNA proofreading in replication. If that mismatch persists after replication, then we have a system called MMR that removes it. MMR stands for mismatch repair. If it persists longer than into G2, then it is removed by BER. BER can actually occur in all of the stages of the cell cycle. MMR always occurs in S phase and very early G2. BER repairs base pair problems. It can repair single nucleotide mismatches from a misincorporation, or it can take out a base that has something added to it that it shouldn't have there, like a methyl group or a bromine or a chlorine. So that's all done by BER. Note that BER specifically repairs changes in the DNA that are smaller than a whole nucleotide. Nucleotide excision repair, on the other hand, also occurs in all stages of the cell cycle except from M phase. Because it works in most stages of the cell cycle, the system is probably the most active for repairing our DNA. So if it's larger than a single base, it is always removed by nucleotide excision repair. If it is a broken DNA, like a single-strand break or a double-strand break, this may induce one of these 3 repair systems. If it's just been broken and nothing has been lost, then we simply ligate the DNA. To repair double-strand breaks is actually this last one called homologous recombination. Homologous recombination with large homology is what sister chromatids do in prophase. This is that crossing over that you've heard about in prophase. If the sister chromatids have already come apart, we often use the method called microhomology end-joining. Microhomology end-joining repairs breaks in the DNA when the nearest thing that we can use to find something to repair it with is a piece of repetitive DNA. So this type of repair typically uses repetitive DNA as the site of repair. Lastly, we have the one that's called non-homologous end-joining. Non-homologous end-joining is the type that repairs when the double-strand break occurs outside of metaphase. Base excision repair and mismatch repair are very highly related. They are color-coded here for which one goes which way. In mismatch repair, we use 2 proteins that are called MUT-S and MUT-L to recognize and isolate the mismatch. Glycosylase will actually remove the base off of a single base pair, so the base is missing. Then endonucleases recognize that as a mistake. The endonucleases come in, cleave the DNA on either side of the mistake, and repair it. The only difference between short patch repair and long patch repair is which enzymes are helping to do the work. In base excision repair, we almost always only fix mistakes that are smaller than a whole base pair. Nucleotide excision repair is the one that does the lion's part of all the repair in your cell. It is the most active repair system for preventing disease. Nucleotide excision repair is the repair system that takes out more than one base pair size repairs. It is your most active form of repair because it actually, in many cases, rides around in its recognition system riding on RNA polymerase II. Notice RNA polymerase II carries the repair enzymes on it. It can also use something called XPC to recognize the lesions around the cell. That's the major difference between transcription-coupled and global nucleotide excision repair. Single-strand breaks, as I've already said, are repaired by simply ligating the gap if it's just needs a ligation, or if there is several base pairs missing, we'll use DNA polymerase beta, delta, or epsilon to actually fill in the gap and then ligate it. Double-strand breaks are corrected by three methods: non-homologous end joining, microhomology end joining, and homologous end joining. They are shown here. The big difference from them is whether or not we have sister chromatids around to use. Notice in homologous recombination, we use the sister chromatid to actually correct our DNA. In microhomology end joining, we actually use a piece of DNA, typically from a repetitive DNA sequence like an ALU or a line sequence, to align our genes as best we can so that we can repair them. Non-homologous end joining is the most gross change in the DNA when it occurs. Non-homologous end joining usually occurs when there is an end of the DNA that has not been repaired and does not have a telomere T-loop on it. Then we recognize those breaks with a special set of proteins. Those proteins then recruit a series of other proteins that eventually result in the ligation of two ends together. Does that end have to be the part that it was broken from? No. And because it doesn't have to be the part it was broken from, this is the major way in which we get chromosomal translocations. Now, the consequences of poor repair is that you get mutations. They are either dominant or recessive. You have learned that with Dr. Larson. If they happen in your somatic cells, not your eggs and sperm, typically just that region undergoes apoptosis, and then the cell will actually be taken care of. In rare occasions, these somatic mutations may accumulate and you may get something like a cancer. Sometimes these mutations cause a gain of function. Gain of function, as you've learned from Dr. Larson, causes an overexpression of the protein, or it causes a change of function of the protein that gives the protein an extra bit of activity that it didn't have before. Loss of function comes in multiple types. You have general loss of function. You also have haploinsufficiency and dominant negative. The primary difference between haploinsufficiency and dominant negative is that in haploinsufficiency, we're talking about something that is a single copy gene, usually, and there is too low a dose of that gene in a heterozygote individual. In a dominant negative, you will always note it is a dominant negative because the protein has to at least dimerize. So homodimers or a multimer are the most common proteins to have dominant negative effects. If they do not invoke that this is a multimeric protein, the loss of function is most likely haploinsufficiency. If it is a multimeric protein, it's nearly always dominant negative. So here are the diseases that are caused by faulty mutation repairs. You do not need to know all the proteins in them. That is just so you know. You do need to know the basic characterization of what happens in these diseases and you need to know which repair system is broken. Nucleotide excision repair diseases are xeroderma. This disease is due to a mistake in global nucleotide excision repair. Notice all nucleotide excision repair diseases cause the individual to become highly photosensitive. Likewise, with double-strand break diseases, there are three diseases. Notice these diseases are predominantly non-homologous end-joining diseases, and they are the most common to cause disease. Homologous recombination usually repairs the gene without causing disease. Microhomology may or may not cause disease. Thank you.